How can you visualize the complex 3D DNA double helix structure? Imagine holding the blueprint of life in your hands. Now picture how scientists turn that tiny, spiraling code into a visual form you can see and understand. Visualizing the three-dimensional shape of DNA has been a journey through many clever techniques. It all started with X-ray diffraction, a method where DNA crystals are bombarded with X-rays. The pattern that appears on photographic film reveals a cross-shaped image that hints at a helical structure. This discovery was key to understanding the double helix shape of DNA. It was made famous by Watson and Crick, who used this pattern to build their model. But seeing DNA directly in its natural state requires more advanced tools. Transmission electron microscopy, or TEM, is one such method. It uses a beam of electrons instead of light to produce images. Scientists can prepare DNA samples in ways that make them visible without staining. With TEM, they can observe DNA strands at near atomic resolution, revealing their shape and how they twist and fold in space. This technique needs specialized equipment and careful sample prep, but it provides a clear picture of DNA's structure. Another powerful tool is atomic force microscopy, or AFM. Think of it as a tiny finger that gently scans the surface of a molecule. AFM physically traces the surface of DNA molecules, creating detailed three-dimensional maps. These maps help scientists see how DNA folds and interacts inside cells. They can even observe tangled regions or loops that influence gene activity. Combining AFM with computer software and artificial intelligence makes it possible to visualize DNA's complex shapes in great detail. Scientists also use staining techniques to see DNA fragments in the lab. Agarose gel electrophoresis separates pieces of DNA by size, and special dyes make these fragments glow under ultraviolet light. While this method doesn't show the three-dimensional shape, it's essential for analyzing DNA in research and medical tests. More recently, computer models have become a key part of visualization. These models take the sequence of bases, adenine, thymine, cytosine, and guanine, and turn them into digital images. They help us see the double helix into and 3 dimensions, making it easier to compare different sequences or understand mutations. All these methods, from X-ray diffraction to electron microscopes, atomic force microscopes, staining, and computer models, have helped us see the DNA double helix in detail. They've driven discoveries that led to modern medicine, like gene therapy and personalized treatments. Visualizing DNA's structure isn't just about seeing a shape. It's about understanding how life works at its most fundamental level.